Mature red blood cells, there's no mitochondria. And therefore there's no electron transport chain, and there's no use of oxygen for metabolism. There's no O2 in metabolism. Most of the metabolic pathways have actually been removed. There's no Krebs cycle. And that's an important feature here, that all the oxygen that is arriving in those red blood cells that is just being used to fill up the hemoglobin. It's being used for transport. The oxygen is not being used for metabolism in the mature red blood cells. They're essentially a bag of hemoglobin with the other organelles missing. And we talked about a particular mutation that exists, the sickle cell mutation, in which the hemoglobin molecules with that mutation will form fibers. And that's because in place of the glutamic acid on the exterior of the molecule, it has a nonpolar valence. Now there is a sort of important fact. Normally this would be something I would test you on, but it now becomes an important fact that it's the deoxy state in which that residue is exposed on the surface, and there's a nonpolar patch, a sort of a depression, or a little valley that's exposed in the surface of the deoxy state. So those then pair up with each other and make this fiber. So that's going to be an important part of our story this morning, and it's the fibers form in the deoxy state when the red blood cells are low on oxygen. Now I'm going to show you a slide that shows a correspondence between regions of the world where this gene occurs in the population and regions of the world where malaria is present. So red alone is sickle cell gene, blue alone is malaria, and then the mixture of those two colors, red and blue, makes purple. Purple is where you have both. And you'll see there's a very strong correspondence between these two features. The presence of the disease and the presence of this mutation in the human population. So it's been speculated that there's probably some reason for this, that perhaps this gene might give an advantage. I mean, there's a selective advantage in evolution here. Why is that gene stuck around? You know, it's harmful. The sickle cell gene gives people sickle cell anemia. But what maybe there's some benefit for that gene in areas where there's malaria. Now the next slide is a very, very busy slide. All kinds of information, and you don't need to know all of this. What should you know? You should know that the malaria organism lives inside mosquitoes and humans. And there's lots of different parts of its life cycle. So, in part of its life cycle, it's in the mosquito, and then the mosquito in the tropics will, bite the human being and infect the human with a stage of that malaria organism. In humans, the organism lives in two places. There's two different stages. One of those is a part of the life cycle that occurs in our liver, and part of it is a life cycle that occurs in our blood. And then in the blood, when a mosquito is taking up our blood, maybe an uninfected mosquito takes it up, it acquires that organism. So there is a portion of this where the malaria organism lives inside our red blood cells, the erythrocyte cycle. Now, when it's inside those cells, it's protected from our immune system. It's not out there in the fluid portion of the blood where our antibodies also are located. It's hiding out in our red blood cells. And this is then the story that's important for what's going on here. Human beings who live in the tropics, who have the normal hemoglobin, called the hemoglobin A gene, the adult genes, they do not have sickle cell anemia. They could get malaria. But they don't have the blood disease. Human beings with both copies of the hemoglobin S gene, that's for this one with this nonpolar valence on the surface, those can get malaria and sickle cell anemia. So they're really bad off. Now what happens if you have a human being that has just one copy of the mutant gene? That's called the sickle cell trait. Not the sickle cell disease, but if you're carrying one copy of it, so half of your hemoglobin molecules are the mutants and half are the wild type. And in uninfected red blood cells, that amount of hemoglobin S does not give sickling. It's not enough. You need all of your proteins to be that mutation, sort of a double dose of that bad gene, in order to have the sickle cell anemia. But the trick here is that the malaria parasite uses oxygen for its own metabolism. It has mitochondria in it. It, is reproducing and going through these various life cycle stages, and it has aerobic oxidation metabolism to make ATP. And so that parasite drives the concentration of oxygen very low inside the infected cells. So in those conditions where you have the extra low oxygen caused by the malaria and only one copy of the bad gene, those two factors alone then will give sickling. It will give sickling. So you sort of need two factors. Either double genes to give sickling or lowered oxygen and one copy of the gene for sickling. Now, so some of the red blood cells, namely those that are infected with the malaria parasite, they will sickle and be these long shapes that can't go through the capillaries and will be broken and will be destroyed by the body. And that opens up the parasites. Now they're exposed to the immune system, and the body can attack them and get rid of the parasites in the infected person. So this is the advantage to having one copy of that gene. It helps you fight malaria because the infected red blood cells are the only ones that will be sickled. So it's an interesting case where something sort of partially bad, or a gene that could be bad if you had two copies, has an advantage if you have only one copy. It's not the only disease like this. There aren't that many. I've run across 2 or 3 of them in my studies. And this is probably the most famous story. So those are my extra comments on medical aspects of